Good afternoon, everybody. My name is Evgueni Ivakine, and I'm from Kids Hospital in Toronto, Canada. Thank you, organizers, for inviting me to give this talk. And today, I'm not going to talk about maps. That some people are showing very beautiful maps and student amino acids, responsible student phenotypes. But what I'm going to do, I'm going to present an approach that I think is going to be very helpful to understand the effect of variants in genes that are highly similar, but also can be applied to pretty much any genes that you're interested in. And I feel like this technology is kind of a marriage between traditional MAVE approach, where you put a cDNA-based sequence in a landing pad system and analyze the effect of variants, and saturation genome editing. So it's a marriage of both. But before I go into talk, just my lab in general is interested in rare diseases, and we are often looking for trying to identify new genes responsible for certain disorders. We are then modeling them in cell lines, in animal models, trying to understand effect, developing genome editing strategies to correct mutations, and then seeing if we can bring it to humans. Today, I'm going to talk about rare disease discovery and variant effect. So it all started from a patient that the family was very interested to understand what's going on, because daughters wanted to have kids and whether this particular disease is going to be inherited or not. So they've done whole exome sequencing, could not find a variant, so they asked our lab to take a look. And what's interesting about the patient, that it was shown to be a classical molar 2 sign on MRI. And basically, if you see the sign, it means it's a Joubert syndrome. So we have looked for genes responsible for Joubert syndrome, could not find any indication that that is the case. So we've done whole genome sequencing for that individual, and what we identify, it's a mutation in a tubulin gene. So it's a glycine to serine change in position 308 in the tubulin gene. And at that point, mutations in tubulin has never been associated with Joubert syndrome. So I reached out to pretty much everybody who worked on Joubert syndrome, and they told me that I was wrong, so it cannot be the case. So that was the first, so they've never seen it. But nevertheless, I guess we found it, and the question was, does mutation in the tubulin gene affect primary cilia? So I'm going to show a few slides, a couple slides about primary cilia, just to remind you. It is probably an organelle that is understudied right now, but it is gaining interest, and it is, you can call it, it's a signaling hub in a cell. So there are many receptors on cilia that go on to transmit signals that leads to certain effects. And mutation affecting cilia development leads to a disease cluster called primary ciliopathies. And Joubert syndrome is just one of those disorders. Right now, about 40 genes, and probably even more, is known to affect Joubert syndrome, but still about 30% of patients do not have diagnosis. And currently, there is no treatment for that. And since we found a mutation in a tubulin gene, just again to remind you that microtubules is basically it's a cytoskeleton of a cell, and it's also molecular pathways. So that's how proteins are going to be delivered to their destinations. So there are alpha tubulins and beta tubulins, and type B is one of the beta tubulins. And there are 10 genes in the human genomes that encode beta tubulins and 10 genes that encode alpha tubulins. So we still, and they're extremely similar, so we still do not know how particular alpha and beta tubulins interact with each other, like alpha 1 with beta 2 and so on. So this stuff is unknown because of extreme sequence similarity between protein families. So, and mutations in the tubulin genes are known to be associated with a class of diseases known as tubulinopathies. And you can see some of the phenotypes that you can find in those patients. And they also have a very distinct presentations on MRI. So anyway, back to our original question. So we find a mutation in a tubulin gene. It looks like it's a Joubert syndrome because of the molar 2 sign. The question is, does it affect primary cilia? And when people are asking this question, we can use fibroblast from a patient and culture it. Well, first of all, we have to confirm that the particular mutation is expressed on at least on mRNA level at the same level. So both wild type allele and a mutant allele are expressed at the same level. And then we can induce ciliagenesis. So this one is a beautiful looking cilia, just using starvation. So media doesn't have a lot of serum. And when you count, basically, you're just counting number of ciliated cells in a dish. And what we're able to show that there's a significant reduction of ciliated cells in patient fibroblasts. But it does not affect cilium length. So basically, again, thinking about, people can say, okay, have we been wrong? So is there actually different genes, maybe in intronic region of some other genes that we were not able to identify by whole genome sequence? So we wanted to make a model for that particular patient. And I said, okay, great, let's make a model. So that is the conservation on the protein level, amino acids, and on the DNA level from multiple tubulin genes that are pretty much identical. So if you want to put your guide somewhere to be able to install a mutation, it's not trivial, either by HDR or by prime editing. It looks like Greg figured it out for a syndrome that was just presented before, but we wanted to think about, is there a different way to do it? So first of all, we wanted to see whether cells, if we remove one copy of type B, whether cells and the cell models that we are going to use, RPE cells, are going to ciliate or not. So we removed one copy of type B, we look at ciliation, and basically it's not affected. So it's not a haploinsufficiency in our case. One copy of type B is sufficient for normal ciliagenesis. And that is the core of the method that we decided to apply. So we used prime editors essentially to install a landing pad, and it's a classical landing pad. We replaced exon 4, where the mutation was, and replaced it with a landing pad for BXB1. So the second allele is still wild type, and another allele right now has a landing pad. And we made a mini circle that contains exon 4 and all intervening sequence with an ATTP site. And in the presence of BXB1 right now, we can simply knock it in. So this one, there's no foreign sequence here except for small scars that are going to be formed, ATTL and ATTR. So we are putting back exon using mini circle technology. And you can make mini circle multiple ways. What we've done here, that's a parental plasmid that you can put your ATTB site, type B, and so on. We can induce recombination between, that's another recombinase, 5C31, and it is quite straightforward way to make a mini circle and a plasmid. And this one can be produced in large quantities. So again, that's basically our new approach, that we can remove exon 4. Tubulin is actually, type B is not a big gene. In principle, we can remove the whole gene if we wanted to, create a landing pad system, create a mini-circle, and recombine a mutated exon 4 into the landing pad. So just to show you a few examples, so we've done it for a few mutations right now, including G308S. So we always check by ddPCR, that's our original parental line, only one copy of type B is present, all of our mutant and also wild type control we put back, 2 copies as expected. Importantly, small scars that are generated in introns do not influence expression of the mutant allele that you can see. It's 50-50 ratio. And right now, we also created a tool that we can study biology of type B, because we can put a very small tag, alpha tag, on a G308S mutant. And right now, we can use normal Western blot to actually detect it. Right now, when you're using tubulin as your loading control, you're actually looking for all tubulins present in a cell. So what do we see? So we compare a G308S clone with ciliation, and it's clearly reduced ciliation in those clones. That mutant, E108K VNU, it was a milder case, talking to clinicians, and the ciliation is reduced, but not to the same degree. And we can start addressing biology of type B. So here, when we immunoprecipitate with tagged wild type, we can clearly detect alpha tubulin and one of the chaperones coming down with the tubulin, with the type B. But when we put a mutant one, we are not seeing alpha tubulin or chaperone coming down. So we are right now trying to understand what's happening. Starting to look at additional phenotypes, and this one is a simple scratch assay that you scratch a well and you wait. You count like how fast it's going to be repopulated with cells, and it clearly that our mutant does not repopulate the scratch very well. It's very different from that. And another interesting thing that we noticed that G308S mutant, they are smaller. So they have a smaller size. So that's one of the phenotypes that we're right now trying to validate and follow up. And there are so many more things that we right now can do with this approach. There are multiple non-pathogenic mutations that we can right now very rapidly to make clones of those and understand their function. We can start looking at interactome of type B that previously have not been possible. And also we can induce degradation using Degron technology and actually see how fast cells are going to react when you degrade tubulin in cells. So overall, right now you can also see that this approach is scalable to saturation. So as soon as you generate in your cell that contains a landing cell line, foundational cell line containing a landing pad system, you can generate a library of mini circles and knock it in. And because it is directional, only one mutant is going to integrate. It's a classical MAVE approach. And then you can analyze those clones. If you know if the phenotypes is, like here, for example, for wound, it can be an arrayed screened, or if the cells are smaller, you can use a flow assay to, for example, sort large cells and small cells to quickly understand effector variants. So there are multiple gene families that can be right now interrogated by that approach. And I also can see that it can be applied to pretty much any gene if you design foundational cell line. Thank you very much. And I wanted to thank people who've done all the work. Thank you.